Hello guys, welcome back to my channel Learning Biophysics with Team 320. My name is Harsh and today I will be talk about cooperative and properties of enzymes. Cooperativity is a phenomenon observed in certain enzymes where the binding of a substrate molecule to one active site influences the affinity of other active sites for the same substrate. This can result in either an allosteric activation or allosteric inhibition of enzyme. Now, types of cooperativity. Positive cooperativity. In this, binding of one substrate molecule increases the affinity of other active sites for the substrate. This can lead to a sigmoidal shape on a substrate cooperation versus reaction rate graph. Examples include hemoglobin and certain enzymes involved in metabolic pathways. Now, negative cooperativity. In this, binding of one substrate molecule decreases the affinity of the other active sites for the substrates. This leads to a hyperbolic shape on a substrate concentration versus reaction rate graph. It is very less common, but some examples are like certain regulatory enzymes. Now, allosteric regulation. Allosteric enzymes have a regulatory site separate from active sites. Binding of an effector molecule, allosteric modulator, to this regulatory site can cause a conformational change, affecting the enzyme's activity. This can result in either positive or negative cooperativity. Significance of cooperativity. Fine tuning of enzyme activity. Cooperativity allows for precise control of enzyme function in response to changes in substrate concentration on effector molecules. Metabolic regulation. Cooperativity plays a crucial role in metabolic pathways, ensuring efficient utilization of substrate and products. Adaptation to environmental conditions. Enzymes exhibiting cooperativity can respond to changes in the environment, such as temperature or pH. For example, hemoglobin, a protein involved in oxygen transport, exhibits positive cooperativity. The binding of one oxygen molecule to a heme group increases the affinity of other heme group for oxygen. This allows hemoglobin to effectively load oxygen in the lung and release it in the tissue. Now, mechanism of allosteric regulation, concentrated model. It is also called Monod-Weyman Chain-Jouet's model. In this, we assume that all subunits of an enzyme exist in either a tense or relaxed state. The binding of a ligand shifts the equilibrium between these states. Positive cooperativity occurs when a ligand binding stabilizes the relaxed state, and the negative cooperativity occurs when the ligand binding stabilizes the tense state. Now, sequential model, also known as Coastland-Nemeth-Filmer model. He proposed that the binding of a ligand to one subunit induces a conformational change in that subunit, affecting the binding affinity of neighboring subunits. This model allows for a more gradual transition between the tense or the relaxed state. Some examples of allosteric enzymes are aspartate transcarbamylase, a key enzyme in the biosynthesis of pyrimidines. This exhibits positive cooperativity with respect to its substrate, aspartate. The binding of a regulatory molecule, aspartate transcarbamylase, inhibits the activity while the binding of ATP activates it. Phosphofructokinase, a key enzyme in glycolysis. This exhibits positive cooperativity with respect to its substrate, fructose 6-phosphate. The binding of ATP can both activate and inhibit this enzyme, depending on the concentration of other metabolites. Hemoglobin. As mentioned earlier, Hemoglobin exhibits positive cooperativity with respect to oxygen binding. This cooperativity allows the hemoglobin to efficiently load and unload oxygen in response to changes in partial pressure. Role of cooperativity in metabolic regulation. Cooperativity plays a crucial role in metabolic regulation by amplifying the response of enzymes to changes in the substrate concentration. Positive cooperativity allows enzymes to rapidly increase their activity in response to small changes in substrate levels, ensuring efficient substrate utilization, providing a mechanism for feedback inhibition. Negative cooperativity can be used to regulate metabolic pathways through feedback inhibition, where the end product of a pathway inhibits an earlier enzyme in the pathway, preventing the overproduction of intermediates. Integrating metabolic pathways. Cooperativity can help to coordinate the activities of different enzymes within metabolic pathways, ensuring the overall flux of metabolites through the pathway is balanced. So that's all for this topic. Now I will complete my next topic in next video. Now my next topic is myoglobin and hemoglobin. Myoglobin and hemoglobin are two important proteins involved in oxygen transport and storage in vertebrates. They share a similar structure consisting of globin protein chain and a heme prosthetic group containing an iron atom. However, they differ in their functions and properties. Myoglobin. It primarily serves as an oxygen storage protein in muscle tissue. It is a single polypeptide chain with a single heme group. It has a very high affinity for oxygen, allowing it to efficiently store oxygen in muscle cells. It releases oxygen to muscle cells during periods of intense physical activity. Hemoglobin. Hemoglobin also transports oxygen from lungs to the tissues and carbon dioxide from tissues to lungs. It is a tetrameric protein consisting of 2 alpha and 2 beta subunits, each containing a heme group. Oxygen affinity of hemoglobin is also very high, but binding of one oxygen molecule increases the affinity of other heme group for oxygen. It ensures the efficient oxygen delivery from tissues to the tissues from lungs and carbon dioxide from tissues to lungs. So now role of I will talk about the different types of curves we have in hemoglobin and myoglobin. So we can talk about hyperbolic shape. Myoglobin has a hyperbolic oxygen binding curve. It is a very high affinity for oxygen, so it shows this curve. Hemoglobin shows sigmoidal shape. Hemoglobin exhibits a sigmoidal oxygen binding curve due to its cooperative binding properties. The role of cooperativity in hemoglobin's oxygen binding. Cooperative binding. Hemoglobin exhibits a positive cooperativity, meaning that the binding of one oxygen molecule increases the affinity for other heme groups for oxygen. This allows hemoglobin to efficiently load oxygen in the lungs and release it in the tissues. Sigmoidal oxygen binding curve. The cooperative binding of oxygen to hemoglobin results in a sigmoidal shape on a plot of oxygen saturation versus oxygen partial pressure. This curve allows hemoglobin to effectively deliver oxygen to tissues over a wide range of oxygen partial pressures. Now the Bohr effect. The pH influence. Bohr effect describes the decrease in hemoglobin affinity for oxygen at lower pH and vice versa. Physiological significance. The Bohr effect facilitates the release of oxygen from hemoglobin in tissues, whereas when carbon dioxide levels are high or pH is lower. This helps to ensure that oxygen is delivered to the tissues where it is most needed. the structural basis of myoglobin and hemoglobin. Both myoglobin and hemoglobin share a common globin fold consisting of 8 alpha helix connected by short lobes. The, the heme group, a porphyrin ring containing an iron atom, is located in hydrophobic pocket within the globin field. The iron atom binds oxygen reversibly. Hemoglobin has a tetrameric structure consisting of 2 alpha and beta subunits. Myoglobin is a monomer. The physiological significance of myoglobin and hemoglobin. Oxygen storage and transport. Myoglobin stores oxygen in muscle cells while hemoglobin transfers the oxygen from lungs to tissues and carbon dioxide from tissues to lungs. Tissue oxygenation. Myoglobin plays a crucial role in providing oxygen to muscle cells during periods of intense physical activity. Hemoglobin ensures that oxygen is delivered to tissues throughout the body. Carbon dioxide transport. Hemoglobin also plays a very crucial role in transporting carbon dioxide from tissues to the lungs, primarily in the form of carbamate. So that's all for my topic. Thank you.